For the first couple of experiments for Lab 1 in Bio10004, we're going to be examining the principles of diffusion and osmosis. So for the diffusion task, we have two chemicals. We have methylene blue and potassium permanganate. Now importantly, methylene blue has a larger molecular size than potassium permanganate. That's the variable that we're altering or manipulating in this experiment. So what we will do is we will take a small amount of methylene blue on a spatula and add it to the center of a petri dish with agar. We'll do the same with potassium permanganate as well. But once we've completed that initial task, we take a baseline measure of the diameter of the coloured blob. We do that using a pair of calipers. So just rotate the petri dish around so that you can take the diameter of the blob from the widest point. And as carefully as you can, take a diameter measurement. In this case, the methylene blue blob from the widest diameter is 17 millimeters wide. That would be your baseline. What you'll do, you'll leave those chemicals alone, and every 15 minutes you'll come back and take another diameter measure. This allows you to see how fast the chemical blob is spreading through the agar. So what you'll do over time is essentially subtract the measurement from the previous attempt from what you've just done to see the difference over time. That is going to show the rate of diffusion, the rate of spread changing over time. And that should be different depending on the molecular size of the chemical. For the second experiment for Lab 1, we're going to be looking at osmosis. Now, osmosis is just a special variant of diffusion, essentially for water movement across a semi-permeable membrane to eliminate a concentration gradient of a solute. So in this basic experiment, we have two conditions. Both conditions are a small amount of glucose mixed with water. The first condition, coloured green, is 5 grams of glucose to a 100 ml of water concentration. The second, in blue, that is 20 grams of glucose per 100 ml of water. So, like with diffusion, where we had a difference in molecular size, we have a difference in concentration gradient this time. And that's what we're manipulating for the osmosis experiment. So these little cells with the glucose solution will be pre-made before you come to class. All we're doing is essentially weighing the cell initially, so carefully weigh each cell. That gives you the baseline weight for each condition, 2.93 grams. Once you have weighed the cell, you are just submerging it in a beaker of plain tap water for a period of 15 minutes, just like in the first experiment. Now, when the 15 minutes is over, you carefully remove the cell, dab dry with a tissue, and be very careful how you do this, just so you don't cause any leaks. And then re-weigh the cell to see how much it has changed over time. Here we have 3.06 grams. And once you're done, re-submerge for another 15 minutes. For the second experiment in the first lab of Bio10004, we're going to be examining differences in body temperature, so specifically surface temperature. We do that using the small infrared thermometers that are provided in class. They look like this. Very simple to use. All you have to do is basically point and pull the trigger on the device. That'll give you a red dot, and all you're doing is aiming the device to the point of the body that you want to take the temperature measurement from. So, I'm going to take a measure of surface temperature from the palm of my hand. As you can see, the temperature on the palm of my hand is about 31.4 degrees. So in your notes, you actually have a list of different parts of the body to take temperature measurements from. Follow those notes, take a measure of temperature at each spot at rest, and then re-perform the experiment after your participant has done some very gentle exercise, perhaps walking up and down the stairs of the CH building. So as part of activity 2, we're also going to be looking at temperature differences around the lab. We're doing that using a thermal camera. So the thermal camera works on detecting infrared radiation. So the warmer an item or object or person is in the lab, they will emit more infrared radiation that gets color-coded by the device as a warmer color. Yellow, orange, red for warmer colors, purple, blue, and so on for cooler colors. It's very simple to use. All you have to do is, once again, aim the device and pull the trigger. And you get a representation of color. So if we just take a look around the room, you can see some basic temperature differences, including my face. Have a look around the lab. Look at your partners. Does a facial covering such as eyeglasses or a cloth mask, does that change the representation of the heat pattern of your participant's face? What about seats, computers, air vents, windows around the room? What's the temperature measure of those different items? For activity 3 of the first lab for Bio10004, we're looking at the electrooculogram. So the electrooculogram, or EOG, is the trace or signal of a method called the electrooculography. Essentially, this is an indirect method of inferring eye movement. So the signal that we're collecting is actually based upon the physiology of the eye. There is a potential, a voltage potential, that goes from the front cornea to the back or fovea of the eye. Our electrodes on either side of the eye will pick up that potential, and that is how we infer movement. If the signal becomes more positive, the eye has shifted gaze towards the positive electrode. If the signal has become more negative, the eye has shifted its gaze to the negative electrode. It's as simple as that. As I said, it's an indirect measure. It's not a direct measure, but nonetheless, we can still infer the shifting gaze of our participant. So the first thing we're going to do for the EOG task is identify where we're putting our electrodes. So we have 2 recording electrodes and 1 ground electrode for this setup. On our participant, the ground electrode will go in the middle of the forehead, and the recording electrodes will go on the temples. So either side of the lateral canthus, that's the outer corner of the eye, on our participant. So the positive electrode will go here on the left, and the negative electrode will go here on the right. Before you attach your electrodes, the first thing you need to do is clean down the electrode attachment sites. Please check out the previous video outlining how this is done correctly. So now we have our participants set up with the electrodes for the EOG. As we indicated before, the ground is in the middle of the forehead, the positive electrode is here on the left temple, and the negative electrode is here on the right temple. So our first task we're going to do is essentially just examining basic saccadic eye movements, that is movements where the eye is shifting gaze from one location to another. So to do this task, our participant looks at the computer monitor, and all they do on instruction from the demonstrator is look in the middle of the screen, look to the left corner, look to the middle, look to the right corner, and so on until we stop the recording. Okay, so look in the middle of the screen for me. Now look left, middle, right, middle, left, middle, right, middle. So as you can see here on the screen, when our participant is looking straight ahead, so both electrodes are equally picking up the potential that runs through the center of the eye. That is when the signal is somewhat middling. Now, when we see a large voltage shift in the positive direction, such as here, because the positive electrode is located next to the left eye, we can tell that our participant has shifted their gaze to the left. Hence, that potential goes positive because the positive electrode on the left is picking it up. The signal then goes somewhat flat because the participant is now looking in that particular direction. They've fixed their gaze to the left. Now, when they shift more rightwards, in this case from all the way left back to the middle, the electrode or the signal becomes more negative again. Hence the signal goes down because now we're moving more away from that positive electrode and more towards the left over on the right. The signal goes flat again as we're looking straight ahead like we did initially. And then the signal goes even more negative because now we've shifted our gaze even further to the right and closer again to that negative electrode. Once again, the signal goes flat as we're focusing our gaze on a particular spot towards the right, and then the signal once again goes positive as we move away from the right, move away from that negative electrode back towards the center. Each of those large voltage changes, that is your saccade. That is your rapid eye movement from one location to the next. So the next task we're going to complete with the EOG is examining smooth pursuit movement. So that's a nice rhythmic, smooth shift in your gaze as you track an object moving in the environment. The object that our participant is going to be tracking is just a key on a string, a very simple pendulum. So our participant will watch the key on the string as it moves back and forth in their visual field. They won't be moving their head, just shifting their eyes. Okay. So as you can see here on the computer monitor, this is the signal that we get when we do a smooth pursuit task, so when we're tracking a pendulum. Here you can see there is a voltage change in the signal. As we said before, when we go more negative, we're moving to the left. As you can see here, when there's a voltage change that's negative, we're moving more towards the right. That's where the negative electrode is located. And as we swing upwards and go positive, that's when we're shifting our gaze back towards the left, where the positive electrode is. Now, with smooth pursuit, this is the type of movement you get, that nice rhythmic back and forth. You can tell that the eye movements are not as abrupt, so not as steep a vertical change with a normal saccade. Instead, we have more of a nice rhythmic sinusoid type rhythm. So for the third activity, we're going to be examining eye movements. So in this case, saccades, during an everyday activity that you do not think about normally, reading. So in this case, we have two samples of text, some very easy text and some more difficult text. So our participant is going to hold that text directly in front of their face. They're going to then read that text silently in their head, not verbalizing. The reason why we don't verbalize is because the electrodes on either temple may then pick up the muscle electrical activity if they're moving their jaw. So don't verbalize for this task. So our participant will just read this silently in his head, and we're going to see what the EOG trace looks like. So just hold it directly in front. Yep. And go. And on with the hard then. So as we can see here on the computer monitor, the EOG trace, whilst we engaged in reading, looks like a staircase. The reason for this is because in English, when we're reading, we start at the left-hand side of the page. So in our case, we're going to be starting very, very positive in our signal. So here would be the start of a line, the top of the staircase. Each time the signal goes flat, we're looking at a particular word or a cluster of words. We'll then have a small negative deflection in the signal as we're shifting our gaze to the right. So we're heading to the next word along the line. It'll then go flat as we focus on that word, and then negative as we shift to the next, and so forth, making a staircase-like pattern. That is until we need to change lines. That's what this very large positive deflection in the signal is. We've gone from the end of one line, looking all the way left to the start of the next. But what you'll notice sometimes, the staircase takes a reversal. Instead of going downwards, the stair goes up. We can see that an example here, and here, here, but also in other signals such as over here and even here. Now normally when we go from the left to the right, the signal goes negative. In this case, it's gone positive. That indicates that we have looked backwards. So instead of moving right, we've gone left. We've reread a word and then continued on reading the line as normal. Easy and hard text, these may differ with how many times you'll reread certain words. You may get more time focused on a particular cluster of words, hence you might see the flat parts of the staircase a bit longer in certain cases. You might see more negative shifts because you're now having to shift more times through a line. Perhaps you can only capture half a more complicated, longer word at once. And you might see more of these repeats, these reversals, going back to reread a word.